This lecture is concerned with the topics covered in Chapter 21 in your textbook, which is evolution at the population level. Populations, recall from ecology, are all the individuals of a particular species in an area. A population is a gene pool, and populations really are the functional units of evolution. Remember the basic evolutionary definition: change in the genetic makeup of a population over time. Individuals don't evolve, rather populations do. So a gene pool, there's a lot going on there. In sexually reproducing organisms, an entire haploid genome gets passed on by each parent. And it's overwhelming to try to keep track of all the genes at once. So it's very common, especially in introductory biology courses, to whittle it down and talk about one gene, right? Pick a gene, hypothetical or real, and talk about the role that gene plays in evolution and how that gene acts in a population. We have some simple mathematical equations we can use when working with population genetics, and one of them is called the Hardy-Weinberg equation. And you might have learned a bit about this at the tail end of Biology 101. So the Hardy-Weinberg equation is, again, a simple algebraic equation that we can use to calculate the expected frequency of alleles or genotypes in a population. Hardy and Weinberg were two individuals who independently came up with the same ideas around 1908. 1908 was an exciting time in evolutionary biology. A generation of natural historians and biologists had read and digested Darwin's writings about natural selection, and Mendel's work had been rediscovered, and there was this merging of genetics and evolution, what some people called the new synthesis. So Hardy was British, Weinberg was German. Neither really was a biologist per se, but they were both interested in natural history and came up with the same ideas about evolution. Hardy and Weinberg proposed a simple expression: P plus Q equals 1. That is the nucleus of the whole Hardy-Weinberg equation. So in this example, the easiest way to apply the Hardy-Weinberg equation is to pick a trait in a population that is Mendelian. You remember that term? Mendelian means you've got one genetic locus and it has two alleles, and the interaction between those alleles produce two different phenotypes, the dominant or the recessive. So in the Hardy-Weinberg equation, P represents the frequency of the dominant allele in the gene pool. It is a frequency. It is somewhere between 0 and 1. A frequency of 0 means it ain't there. A frequency of 1 means every single allele of the gene is the dominant allele. Q then represents the frequency of the recessive allele. So, of course, P plus Q has to equal 1. You have to account for every allele. If an allele is not the dominant version, it is the recessive version. So think of a pie chart. You got a whole pie, right? If you cut it exactly in two slices, P and Q both equal 0.5. Now, it gets interesting when you expand that expression P plus Q equals 1. That expansion allows you to go from talking about the frequency of alleles to talking about the expected frequency of genotypes. So here we're talking about diploid organisms. They have 2 alleles, so there are 3 possible genotypes. So when we expand that expression P plus Q equal 1, when we square it, we get this. P squared plus 2PQ plus Q squared equals 1. Now what we have there, those 3 expressions are the frequency of genotypes. To have 2 copies of the dominant allele, that's what P squared represents. It is the expected frequency of the homozygous dominant genotype. 2PQ is the expected frequency of heterozygotes. And that leaves us with Q squared as the expected frequency of the homozygous recessive genotype. All right, so how do you actually use this formula in a more realistic setting? Okay, so imagine you're studying a population, and what you see are phenotypes, right? And if you're talking about a trait that's under simple Mendelian genetic control, the dominant phenotype could be one of 2 different genotypes. They could be homozygous dominant or heterozygous. But individuals with a recessive phenotype, you know what their genotype is. They have to be homozygous recessive. So that makes the recessive phenotype a very important known. If you have the frequency of the recessive phenotype, that's your anchor. That's the known you can use to calculate the unknowns, namely the frequency of both alleles and all 3 genotypes. Okay, let me give you an example. I've been studying cave-dwelling snails for a few years now. And this is a picture in Fogelpole Cave, which is Illinois' biggest. It has 15 miles of passage. So there's a major stream flowing through the cave. And in that stream, there are snails. And there's a species of snail there, which has some interesting biology. Some of the individuals are totally void of pigment. They're albinistic, if you will, and that's a pretty common feature in cave-dwelling organisms. But some of them have pigment, and they're part of the same species, so that's kind of an unusual situation. So some colleagues and I did a census. We went into the cave and sampled them and figured out how many had pigment, how many were albino. Then brought them back to the lab, did some crossbreeding between them, determined the albino phenotype is recessive. All right, so we know that's the recessive phenotype. What we found is 67.3% of the population were albino, and 32.6 or 7% of the population were pigmented. So there we have the frequency of the 2 phenotypes. That slice of the pie that is represented by the pigmented individuals could be homozygous dominant or heterozygous. But we know that slice occupied by the albino has to be homozygous recessive. So what we know right now is Q squared. We know the frequency of the homozygous recessive genotype, because you have to have that genotype in order to have the recessive phenotype. Now, to carry on from there, we need to know Q. So you would take the square root of that. The square root of Q squared tells you Q, which is 0.82. 82% of the alleles in the gene pool are the recessive allele. So right away here, here's a misconception you might have taken from Biology 101, that the dominant allele is always more common. Dominance and recessiveness doesn't have anything to do with frequency, right? So a recessive allele can be more frequent, more common than a dominant allele. Because P plus Q has to equal 1, we know that P must be 0.18, right? So 1 minus 0.82 tells us what P is. So we know the frequency of both alleles. If you square P, you know the frequency of the homozygous dominant genotype. 3.2% are homozygous dominant. We know the expected frequency of heterozygotes. 29.5% of the population should be heterozygous if the frequency of Q squared is 0.673. And we already know that last piece, right? So that's pretty cool. I think you can use the frequency of the recessive phenotype to calculate the expected frequency of both alleles and the expected frequency of all three genotypes. Now here's an extension of the Hardy-Weinberg equation. It is a concept called Hardy-Weinberg equilibrium. It is the idea that the frequencies of alleles and genotypes in a population will not change, hence equilibrium, from generation to generation if certain conditions exist. Now, Hardy-Weinberg equilibrium is possible because meiosis is a random process. It is the randomness of meiosis that makes this possible. So here's an example in your book. Imagine we have this population where the frequency of the red allele, which we're calling P, is 0.8, and the frequency of the white allele is 0.2. When the adults in that population go to make their gametes, the males, 80% of the gametes should have the red allele and 20% should have the white allele. The frequency of alleles in the gametes produced mirrors that of the frequency of the allele in the gene pool because of segregation and independent assortment and the randomness of meiosis. The same is true for the females. So if you take a batch of eggs where 80% of them have the red allele and a batch of sperm where 80% chance of having the red allele, and you throw them together, you're going to perpetuate the same frequencies from generation to generation if certain conditions exist. All right, so what are those certain conditions? The first is that population size must be large enough to prevent chance sampling from affecting the allele frequencies in gametes. So when you have a big enough population, small random events will have little or no impact The smaller the population is, the higher the likelihood that a sampling error will occur or that random events will impact the frequency of alleles that get passed on. So our first condition is population size has to be large. When population size gets small, we can have a phenomenon called genetic drift. And one form of drift is called a genetic bottleneck. That's what happens when a population crashes due to a random event. And that's illustrated here. If you have this gene pool is the bottle, and the genes in it are different alleles and they have different colors. And there is a bottleneck event, a crash in the population size. That is akin to tipping that bottle up, giving it a shake. The constriction in the neck of the bottle will only allow a few beads to pass through. That's our sampling of the gene pool that will establish the new population. So that new population will be different than the original. Illinois is used as an example in your book to talk about a genetic bottleneck with respect to a bird called the greater prairie chicken. Illinois, the prairie state, right? It's really the state formerly known as prairie because 99.9% of our prairie is gone. It's got turned into row crop agriculture. And prairie chicken habitat disappeared in a matter of a couple decades. So the remaining populations went through a bottleneck and have very low levels of genetic diversity, and that has had a real negative impact on their reproductive success. So decreases in population size can have profound effects on gene pools. Okay, a second condition needed is that you can't have any mutations, right? Mutations at the gene level. That is how we get new alleles, right? You have an allele, and there's a change in the nucleotide sequence, and now it's a different allele. So you can't have that. If you cause a mutation in a gene, you're changing the frequency of both alleles, right? So mutations cannot occur if a population is to remain in equilibrium. Of course, mutations are spontaneous and random. Third condition, can't have gene flow. Can't have genes flowing in and out of the population. So what we're talking about here is immigration and emigration. If you have individuals joining a population, new genes are flowing in. If you have individuals leaving the population to go somewhere else, genes are flowing out. So gene flow can cause disequilibrium, can cause the makeup of the population to change. Gene flow of a species is affected by several factors, one of them being anatomy. Some organisms have a body type that makes it really hard for them to pick up and move somewhere else. Others might be restricted by habitat if they are specialists to a particular habitat type. I mentioned my interest in caves. So here's a cave-dwelling flatworm that lives in Stemler Cave outside of Colombia. That population is like an island in that cave. Although this species also lives in Illinois caverns and Focal Pole south of Waterloo, the flatworms in Stemler Cave aren't going to pack their bags and go visit their cousins in the south. They represent isolated gene pools. This is a map showing the surface streams in southwestern Illinois. So what I'm talking about here are different cave systems outlined in red, and they are separate groundwater systems where the cave-dwelling creatures represent isolated gene pools. All right, our next condition is that mating has to be random. Random mating means all individuals are equally likely to mate and pass on their genes. No single individual would have more offspring than any others or be more likely to pass their genes on than any other. So it's not enough for meiosis to be random, but mating has to be random. You have to take those gametes that are produced and imagine they're randomly being tossed together. Our last condition is you cannot have selection. You cannot have forces that favor the survival and reproduction of some individuals over others. Natural selection is a primary way that evolution happens. Humans are potent agents of evolution. We guide and direct the evolution of some species through what we call artificial selection, or what I call human-directed selection. Before we move on, I want you to stop and think about something. The Hardy-Weinberg equilibrium is a concept that tells us what does it take to keep a population in genetic equilibrium. Essentially what it's saying is, what does it take to keep evolution from happening? All you have to do is violate one of those conditions, and we will have change in the genetic makeup of a population over time. The frequency of alleles will change from one generation to another. Although the example of the flatworm, for example, they might not be able to go through gene flow. So if that was the only factor, the population might not evolve. But there are other factors. There are selection forces acting on them. Mutations happen spontaneously. The reality is, evolution is constantly happening. There really is no population in equilibrium for any significant amount of time. But this is still an important concept. This concept of equilibrium, what it does is gives us a checklist of the reasons why evolution happens. If you're studying a population in nature and you find that their allele frequencies have changed, now you have this set of hypotheses you can test. You have these 5 reasons why evolution might happen, and among the most important of those is selection. And that's what I want to do next, is talk a little more about selection, particularly natural selection. So natural selection, again, is a mechanism. It is a way that evolution happens. What we see in this illustration here is an example of how powerful selection can be. This pile of vegetables, broccoli, kale, cauliflower, cabbage, they all come from the same ancestral plant. And it's humans that created these by selection. Now again, that's what we call human-directed selection, but it is an analogy, a model for how natural selection works. And in fact, the primary author of natural selection used artificial selection or human-directed selection as a model or a metaphor. So that author I'm referring to is Charles Darwin. And Darwin actually isn't the only person who came up with the idea of natural selection. Independently, another British naturalist named Alfred Russel Wallace also developed the same ideas, but it is Darwin who published a comprehensive argument about why natural selection is a real evolutionary force and is the person we consider the primary author of the concept. So Darwin published On the Origin of Species by Means of Natural Selection in 1859. So I hope you learned something about natural selection biology 101. I'm not going to go into it in a lot of detail here. It is, again, I hope something you learned about, but here's a reminder of a few key concepts. When we talk about natural selection and how it works, we tend to use traits that experience what we call continuous variation. Traits that vary along a spectrum. What you might have spent most of your time on in Biology 1 are examples of traits that have discontinuous variation, like my snails, pigmented or not, or pea plants that are tall or short. Now, those are important, and traits like that can experience natural selection. But when we talk about the subtleties of selection, it's more useful to use examples that have continuous variation. And the reality is most biologically important traits do vary along a spectrum, continuous. Most things that make us human are traits that have continuous variation. So when you graph such a trait in a population, it tends to exhibit a normal distribution, and that's what we see on the right. The highest number of individuals in the population have the combination of alleles that would give you the phenotype in the middle of the range of variation, that so-called bell-shaped curve. All right, so the whole essence of natural selection is this idea that there are forces in nature that we call selection pressures that cause a difference in the rate of survival and reproduction in a population. Natural selection is all about differential survival and differential reproduction. Any individual with a gene variant that allows them to survive and reproduce a little better in the face of that pressure will be favored. Those genes, those traits will become more common over time. Natural selection is not the only way that evolution happens, right? You can have genetic drift, you can have bottleneck events, but natural selection is what causes adaptations. And adaptation is a feature that increases the likelihood of survival. Fitness is another thing that can increase with natural selection. Fitness in the evolutionary Darwinian sense is a measure of reproductive success. You are more fit if your genotype is more likely to be passed on to the next generation. So adaptation and fitnesses are signatures of natural selection. Now, natural selection can play out over several modes, 3 of them primarily. So we're going to talk about 3 different modes of selection. And our starting point here is our original population. So we got what? Population of mice. And the trait that we're looking at on the x-axis is the color of the fur. And the population varies from light to dark, with most being medium, right? So the y-axis is the number of individuals in the population. And we see that the population can respond to different kinds of selection pressures. There are 3 responses, 3 modes of selection. The first mode of selection is the one that we see most often in nature. It's called directional selection. And look at this figure. Here it is visually, graphically. What we see in this figure, again, the original population on top. Below is sort of the after. This is the distribution of the phenotype in the population after directional selection has occurred. The blue arrow that you see, this blue arrow right here, okay, it is showing us the portion of the original population which is selected against. The individuals at that end of the range of variation have lower rates of survival and reproduction. The genes that confer the darker colors are favored under this particular selection pressure. Those genes get passed on at a higher frequency, and the population experiences a shift along that x-axis away from the less fit end of the range of variation toward the more fit end. So as a population, they get darker. More of the individuals inherit the alleles that cause darker fur color, and the population gets darker. That's the adaptation. In this case, being darker, let's say they're living on a dark substrate and it's harder for predators to see them. So remember, natural selection is driven by specific selection pressures, things like predation. This is an example of directional selection that was documented in a group of finches on the Galapagos Islands, which is a significant location because it's one of the places Darwin visited that inspired his thinking. The lighter colored graph shows us the distribution of beak sizes in these ground finches in 1976. There was a drought on the island, and lots of plants failed to make fruit and flowers and seeds. The bird populations were much diminished, but the individuals that had the greatest success at foraging are the ones that had larger beaks because they could crack the seeds of the more drought-tolerant plants that had thicker seeds. They had more reproductive success, and in the next generations, over the next couple of years, a demonstrable shift in beak size happened, right? A directional selection toward having a larger beak. Okay, second mode of selection is called stabilizing selection. An example of stabilizing selection that I think is really interesting is human birth weight. To the degree that the size of a baby at birth is genetic, historically we have been under the constraints of stabilizing selection. In stabilizing selection, the individuals in the middle of that range of variation are the ones that are favored. The genes, in this case, that cause babies to grow at a moderate rate so that they're between 5.5 and 8 pounds is optimal. When you look at this graph and see at low birth weight, mortality goes up. At high birth weight, mortality goes up. So the first one might make more sense than the latter. Very low birth weight babies, their organ systems aren't as developed, their immune systems aren't as developed, they don't have reserves of fat and have nutrients, and yeah, it makes sense that they would have a higher rate of death. So if it was genes that caused them to be small at birth, that would be selected against. But what about the other end? Well, we're talking here about before most modern medical intervention. If you had a baby that grew very large, that puts a tremendous amount of stress on the mother. And they say if mom ain't happy, ain't nobody happy. That's particularly true during childbirth. So genes that would cause babies to get large were selected against because more often the mother would be in physiological stress. The mom might die and or the baby would die. So human birth weight historically has been under the constraints of stabilizing selection. We can think about stabilizing selection from the perspective of single genes. Sometimes being a heterozygote confers an advantage. There's a heterozygote advantage, which to me is really sort of just a version of stabilizing selection. Your book talks a bit about how the sickle cell anemia allele is very common in Africa, much more common than you would think for a gene that causes a serious disease. But if you are a heterozygote, you also have a great deal of resistance to malaria, which is a very debilitating infection. So selection has favored heterozygotes over individuals who are homozygous normal and would be more likely to get malaria, or individuals who have homozygous for the sickle cell allele who has sickle cell anemia, which is a serious disease. All right, here's our third mode of selection called disruptive selection. And it's basically the reciprocal of stabilizing. So in disruptive selection, it's the median genotype and phenotype that is selected against, and individuals at both extremes are the ones that have higher rates of survival and reproduction. So as a result of disruptive selection, we get the evolution of dimorphisms. So a dimorphism is the term we use for when there are two forms or two phenotypes in a population. That previous image showed two birds, one with a big beak, one with a small beak. That's a dimorphism. These are sunfish. These are bluegill, which is the Illinois state fish. Did you know that? We have a state fish. And within male bluegills, there is not just a dimorphism, but a polymorphism. There are actually 3 genetically distinct forms of males. And these are maintained through multiple modes of disruptive selection. And we're going to focus here on the 2 morphs we call the territorial male and the satellite male. So territorial males are what we think of as the normal, typical male. They defend a territory. They're large and colorful. They swim around the edge of their nest to attract females. When females enter the nest, they mate. They have external fertilization, like most freshwater fish, most fish generally. As the female releases her eggs, the male releases his sperm. The eggs are fertilized, the female goes off, the male stays and guards the nest and guards the eggs until they hatch. There's a form of male called the satellite male. I call them satellites because they tend to hang around the edge of the male's territory, kind of circling around like a satellite and waiting until a mating event happens, and then try to slip in. Now, you might think that the male would just chase him away or beat him up. But the trick about satellite males is they mimic the appearance of a female. So the dimorphism here is among males is you look hypermale, in which case you're a territorial male, and you defend a territory and attract females, or you look like a female, and you make a living as a satellite. So in this picture from Natural History Magazine, on the left, we have the male, the territorial male, and then in the center, there's a female. But then we have the satellite male. The satellite male looks very convincingly like a female. And this guy, in his little fishy mind, thinks he's got two ladies laying eggs in his nest. The reality is, one of those ladies is a guy who's releasing sperm and fertilizing some of those eggs. And guess what? You're going to be stuck taking care of them. So that's called being cuckolded. So that's, to me, an interesting example of disruptive selection. And this was discovered here in Illinois by biologists at U of I, that males come in two forms, satellite males and the territorial males. So that last example of disruptive selection involving male bluegills, where the satellite males are exploiting the differences between males and females. Right? The differences between males and females in the species are called sexual dimorphisms. And sexual dimorphisms are created and exaggerated by a type of evolution called sexual selection. So sexual selection is when individuals of a certain characteristic are more likely than other members of the same gender to mate and pass their genes on. So peacocks are an example that's often used of sexual selection. The large tail feathers of males is attractive to the females, apparently. And sexual selection has created the situation where that difference between males and females gets bigger and bigger and bigger as sexual selection favors males with bigger and bigger tail feathers because females are apparently attracted to that. So sexual selection is, I guess, a special case of natural selection where we have exaggerated differences between the genders because of the idea of mate choice. It somehow involves mate choice and recognition of gender between males and females. Now, finally, just a couple more notes about natural selection. Previously, we talked a little bit about convergent evolution. So convergent evolution happens when different species are exposed to similar selection pressures and they evolve similar adaptations. And there are some very striking examples of convergence on broad scales. One of them is in the placental mammals of North America and the marsupial mammals of Australia. So Australia is populated by pouched mammals. North America is populated by placental mammals. And on each continent, ancestral mammals diversified and adapted to different resources and exploited different niches, and many examples of convergences came out of that. So we have, for example, a large predator, the wolf and the Tasmanian wolf, which unfortunately is now extinct. There's a marsupial version of a mouse. We have flying squirrels, they have sugar gliders. Now, the marsupial mouse is more closely related to the Tasmanian wolf than it is to the mouse. The flying squirrel is more closely related to the wolf than it is to the sugar glider because they're both placental mammals. So convergence can happen on really big scales. One last example here. In the history of backboned animals, the power of flight has evolved at least 3 separate times. And that would be in birds, in pterosaurs, which are an extinct reptilian form, not a dinosaur, but a separate lineage, and then in mammals and bats. So we see a lot of convergences in the adaptations in their forelimbs, lightweight skeletons. For the bat, most of the wing membrane is supported by enlarged fingers. For the pterosaur, most of the wing membrane is supported by a great, huge, long pinky. For the bird, it's feathers that give them their surface area. So, interesting convergences when it comes to flight.